Great, so we've talked about the cytoplasm already, and we talked about the non-membranous organelles, which means they're not wrapped in a membrane. So let's move on to the membranous organelles. Whoops, went one too far. So membranous organelles, remember, are wrapped in the lipid membrane. They're the phospholipids that go around just like the outside of the cell. And what happens is that most of those organelles will actually bleb off of other structures. So they have the same phospholipid structure around here. Actually, I shouldn't say most. We'll say some of them bleb off like this, but they have the same membrane on the outside. So for instance, if we have something we're going to refer to as the rough endoplasmic reticulum, it has this structure. When it's ready to ship something, it will pinch off those structures, those little products that it's made, ship them across to another part of the cell where it just blebs together and just merges together with another membrane-bound organelle. So they have the same outer membrane. They can just pinch off one section, send it over to another one, and then merge into another. Same idea happens when we go to take one of these products and we can send it to the outside of the cell. It just ships to the outside of the cell, merges with this membrane, and then pumps away and out of the cell. So let's talk about some of those. The first one's going to be the endoplasmic reticulum. And when we started talking about ribosomes, and we're going to talk about them in more detail now. The endoplasmic reticulum, if you look at a cell, right in the center of the cell is this huge nucleus. And the endoplasmic reticulum actually is continuous with the nucleus. So if you look at the nucleus, it has its own membrane. As it comes around to the outside, it just kind of folds over and then starts forming this area called the endoplasmic reticulum. We can subdivide the endoplasmic reticulum in two sections. We can call one area the rough, and you can see how rough this is because it's studded with those little ribosomes. And we call another area the smooth, and you can see there are absolutely no ribosomes. Well, let's see if you remember from the last video. What do the ribosomes do? Ribosomes make what? They make proteins. So in this situation, this rough endoplasmic reticulum, these are flat sacs that are making proteins. But the smooth has no ribosomes, so what can it not make? It can't make proteins. So it's going to modify things like carbohydrates and lipids. So when you look at it again, the rough ER has this granular appearance where the smooth has a smooth appearance. We'll talk a little bit more in detail. So you have to know the rough endoplasmic reticulum, and I always think of it as a protein factory. So you have this big factory. All of the blueprints, everything you want to make in the cell, the recipe or the blueprints are stored in the nucleus. So let's say that we want to make something. We open up that recipe book, we make a copy of that recipe, and we bring it out here to the endoplasmic reticulum. If I zoomed in on the surface of this endoplasmic reticulum, this is what it looks like. Here you have ribosomes along the surface. So I bring the recipe over, and that recipe will turn into my product, which is a protein. And then the protein gets pushed into the endoplasmic reticulum. So here it says the ribosomes stud the outside of the surface. The RNA, the recipe for whatever you're trying to make, is turned into or translated into a protein, and that protein is injected inside these membranes of the rough endoplasmic reticulum. So now it's inside. Once this protein's inside, you can actually package it in this little tiny thing called a vesicle, and we can ship it somewhere else in the cell. And I'll show you exactly where it gets shipped. Right? So again, here's the rough endoplasmic reticulum. Here are your ribosomes. Remember from the last video, you need these two parts of ribosome to make one functional ribosome. Here you can see the rough endoplasmic reticulum because it's all studded. There are all these little bumps all over the place that are ribosomes. Here's the smooth that still touches the rough, but there are no bumps. So again, ribosomes. The keys you have to remember is that they're made out of two proteins with something called ribosomal RNA, or rRNA is the abbreviation for ribosomal RNA. Right? They can be free in the cytosol, which we talked about before. They're not membrane bound. They're just free floating in the cytosol. Or they can be fixed to the endoplasmic reticulum, which makes the endoplasmic reticulum look rough. Right? And then that protein translation, or the protein production, is called translation. You take a recipe, you translate that recipe into a final product. So again, I always think of these little ribosomes as being the assembly line workers that sit on the edge of the endoplasmic reticulum. Here you can see the endoplasmic reticulum. You can see all those little studded ribosomes along it. Right? If we zoom in and look a little bit closer, here are the ribosomes, and you can see the ribosomes making this long chain of protein. So they make proteins. This is something you'll talk about more in the genetics section. Right, next is a smooth endoplasmic reticulum. The smooth endoplasmic reticulum is continuous with the rough endoplasmic reticulum, but it has no ribosomes, which means it's no protein synthesis. Notice the red words again. You have to absolutely positively know these things. What the smooth endoplasmic reticulum does do is it helps synthesize lipids. So the specific lipids are things like fatty acids, triglycerides, phospholipids that you make membranes out of, and then things like steroids like estrogen and testosterone. Right? And we'll talk about those later. It also helps store calcium. When we talk about the muscle cells, the muscles have something called a sarcoplasmic reticulum. All it is endoplasmic reticulum, but in a muscle, so we changed the name a little bit. But it stores calcium there. And we'll talk about calcium later, how it helps with contraction of muscles. And then another really important thing the smooth ER out here does is detoxify alcohol, drugs, and metabolic wastes. So here's a big point you want to take home with you, or a point you want to remember. When you're looking at these organelles and you're thinking, why the hell do I have to remember all this stuff? There's actually a reason. There's a big picture we're building up to this semester. We're taking all these little pieces of information and making one big idea or leading up to something. If you extract a cell out of the body, let's say you take a cell out of the body and you take it from an organ and you look at it under a microscope, that cell represents the main purposes of what's going on in that organ. So, for instance, if I'm pulling a cell out of the liver, well, what's the main purpose of your liver? It synthesizes lipids, it helps store fats, it helps modify proteins, it helps detoxify alcohol and drugs and metabolic wastes. I don't know if you're noticing a pattern. But every cell in your body has all of the organelles, except red blood cells, but they have different proportions. If I take a liver cell out, what organelle do you think it's going to have a ton of? It'll have a ton of the smooth endoplasmic reticulum. It has a lot of the smooth endoplasmic reticulum because its main job is to detoxify things like drugs and alcohol and metabolic wastes, or to synthesize lipids. So you're going to see a lot of this endoplasmic reticulum inside of a liver cell. That's the point. Well, we're going to talk about, mitochondria in a few minutes, and mitochondria are there for energy. What cells in your body require a lot of energy? Think about when you're moving around, you want to lift something. What kind of cells are requiring lots of energy? Muscles. I talked earlier about how your brain uses a lot of electricity, so it needs to use a lot of energy. It's going to need a lot of mitochondria too. So look at proportions. I'm not just trying to make you memorize everything. I really want you to understand why you need to know these things. It means a lot more and it'll help you remember it if you actually try and understand why. Right, so here you have the nucleus again. Here you have the rough ER and the smooth ER. You can see how they're all continuous with each other. They all touch each other. They're one continuously long structure. We just subdivide it. You have the wall of the nucleus, you have the endoplasmic reticulum, and then you have the smooth ER. Again, when you're looking at it, you can see how rough and jagged this rough ER is, or you can see how smooth and lipidy all of the smooth ER are. Alright, so now we've made some products. I always look at the endoplasmic reticulum like factories. They're making things. Rough makes protein. Smooth makes or modifies carbohydrates and lipids. Now once you've made these products, you need to do something with it. If this were a small town and my warehouse is making the products, I need somebody to package the products, to ship the products. So what I'll do is I'll actually give my unfinished products over to this thing called a Golgi. So if you look at the smooth ER. It has these little structures called vesicles that bleb off, and then they come over here and go to the Golgi. The rough ER will do the same thing. It'll package enzymes, proteins, and ship it over here to the Golgi. So let's talk about the Golgi now. So the Golgi apparatus' main purpose is to modify, package, and ship. I always think of it like the UPS of your little biodome. It's for packaging and shipping, or USPS, I guess, whichever perspective you want. But it's stacks of flattened membranous sacs. So these sacs going back and forth are like pancakes to me. They're kind of wobbly, flat pancakes. Here you can see the electron microscope graph. They're kind of like stacked pancakes, one over another. So what happens is, here's a package coming from the endoplasmic reticulum. It comes over here, but this is full of unfinished goods. It merges, its membrane merges with the Golgi apparatus and pushes all of its products on the inside. They call this the cis face. Cis means it's closest to the rough ER. Right? If you've taken organic chemistry, you know cis and trans. Cis means the same side. Trans means the opposite side. So cis means the same sides of the rough ER. It'll process it, package it, move it back and forth, clean up the stuff. It'll take those proteins and change them. It makes them look different. It makes their structure different. And then finally, when it gets over to the trans face will have, like, for instance, this might have green and purple proteins in it. Over here you'll have just packages of purple. Over here you'll just have packages of green. It sorted them for you. It relabeled them. It put new stickers on this package and then sends them off to wherever you want. So the transface releases these things and they go wherever they need to. If this is something called a secretory vesicle, you're going to secrete it. You're going to exocytose or dump it out of the cell. If this is, let's say, just something for storage. So this vesicle, let's say it stays inside of the cell. It'll be moved somewhere else. Right? So it's kind of a really cool process. You take the rough endoplasmic reticulum, you send the products over here. They go back and forth to these different things called cisternae. Starts at the cis side, it keeps working back and forth until it's sorted completely, and then releases from the trans side. That's the Golgi apparatus. So again, packaging and shipping. And the main function you want to remember is that it takes products from the endoplasmic reticulum, brings it into the cis side, sorts them, organizes them, and releases them from the trans side of the Golgi. What's happening here is something called post-translational processing. Remember, the rough endoplasmic reticulum translates the RNA. So it writes or makes the proteins. But once those proteins are made, this is post-translational. This is like the finishing steps, and that's what the Golgi does. And then it packages those proteins to ship them other places. These are the 3 places you can ship it to well, after you're done. You can either export it out of the cell, so you bring it out here to the outer surface and you dump it out like this. So you can see the products made in the endoplasmic reticulum. They come to the Golgi, the trans side releases it, and moves across, merges, this outer membrane merges with the plasma membrane of the cell, and then it exocytoses this stuff to secrete it. So if this were a hormone, it just dumped it out. If this were like a waste product or something, you could dump it out here too. Another thing it can do is that it can incorporate things into the plasma membrane. And there's no really good picture, but you have to imagine this. So you make something, you stick it in this, like a receptor. You stick it around the outside of this vesicle, and then when this vesicle merges with the outside membrane, because those receptors are stuck in this membrane, they become part of the plasma membrane. So if you're making receptors, let's say receptors for insulin. So type 2 diabetics, they don't have those receptors. What happened is that something in this process failed. Either they couldn't make the receptor, they couldn't code for it with their DNA, they couldn't package it properly, they couldn't release it out here onto the wall properly. Something's going on that's making it not work. So that's 2 things. You can either dump stuff out of the cell, you can make it a part of the cell membrane, or the third is that you can just program to go to other organelles, or you can turn it into an organelle. Like this third thing's called a lysosome here, or this thing's called a lysosome. It's made with enzymes, protein enzymes that came off the Golgi, and now they're just floating around. The lysosome is something called a vesicle, but it's a specialized vesicle that acts like a stomach in the cell, which we'll talk about in just a few slides. All right, so this endomembrane sequence of events, endomembrane, is talking about the process of all these membranous organelles working together. So you take and you start by making the protein into the rough endoplasmic reticulum. You pack it in a vesicle and you bring it over to the Golgi. The Golgi unpackages it, modifies it, shortens it up, puts a new shipping label on it, sends it out the trans side, and then, remember, it does one of 2 or 3 things. It can either exocytose, it can put it in this membrane and just turn this into part of the membrane, or the third thing it can do is turn it into a new organelle, like a lysosome. Right. So let's talk more about those vesicles. I already mentioned one vesicle called a lysosome, but vesicles are just shipping and storage containers. They're like little cargo structures. So here are some examples. You can have a structure that comes off the Golgi that's a peroxisome, and it uses hydrogen peroxide, by the way, is how it gets its name. It can detoxify alcohol and waste. So what other organelle does it work with? What other organelle detoxifies alcohol and metabolic waste? The smooth endoplasmic reticulum. So remember our whole point. What organ in your body will you find the cells having lots of peroxisomes? Well, that'd be the liver again, right? Because it likes to break down things. Another interesting thing is that you can imagine this being a white blood cell. The white blood cells destroy bacteria. They break down toxins. They break down things that invade your body. So they're going to be full of peroxisomes too. Right? So the peroxisomes will take and break apart the alcohol. It makes it safe. The peroxisome starts with something dangerous like hydrogen peroxide, but its final product is actually water and oxygen. So this harmless waste is dumped out in the environment. It just dumps water and oxygen into your environment. This is a very green destroyer, right? So It's green because it doesn't make harmful wastes. Another type of vesicle is a lysosome. The lysosome branches off, and the lysosome is full of hydrolytic enzymes, water-destructive enzymes. So it's an enzyme, just add water to it, and it starts destroying things. So let's say that this white blood cell swallows a bacteria. Well, that bacteria is what? What was the process? Is it pinocytosis? Is it phagocytosis? Or receptor-mediated endocytosis? What's to eat? That would be phago. So the bacteria is phagocytized. The cell goes along, it wraps itself around the bacteria, doesn't punch a hole in the wall of the cell, it just wraps around it. It moves this vesicle, this transport vesicle, over. The lysosome merges with it and makes something called a phagolysosome, or phagosome. That enzymes in here, the hydrolytic enzymes, eat the bacteria. The cell can keep the good stuff, like some proteins and some sugars, and then it can spit the bacteria byproducts out, just like pooping it out the other side by exocytosis. So these vesicular transporters, these vesicles, can move things all over the place around in the cell. All right, let's talk about that process again. So when you're making an enzyme called lactase, enzymes are made out of proteins. They're all made out of proteins. So where would it be made? In the cell, what organelle makes proteins? The rough endoplasmic reticulum, right? So DNA is in the nucleus. It's turned into RNA, the recipe. The RNA moves to the rough endoplasmic reticulum. So here we have the cell. Here's the DNA turned to RNA. It moves out to the rough endoplasmic reticulum, which should be continuous, right? And it makes this big protein structure, this huge protein structure, right? Once it's made it in the rough endoplasmic reticulum, the next thing it's going to do is it's going to ship it over to the Golgi. The Golgi is going to take this large structure and trim it down a little bit. This is too big. So it trims it down into something a little bit smaller. Okay? So they actually call this pre-prolactase. They call this prolactase. And then finally, when it gets into the vesicle is going to convert it into something called mature lactase, or just lactase. And then when you're ready, because you ate some lactose, this lactase enzyme is dumped out of the cell into your GI tract. So it's going to be dumped out into the small intestine, technically. And then it's going to find lactose and break it apart. Right? And we'll talk about this when we get to the GI tract, because here you have all these little enzymes that are stuck to the surface of that cell. When lactose comes along and bumps into it, it breaks lactose apart so you can digest lactose. People that are missing this enzyme are called what? Lactose intolerant, because they're missing the enzyme. Something along this process doesn't work. Maybe their DNA doesn't code right. Maybe the rough endoplasm is not making the protein correctly in the first place. Maybe the Golgi is not packaging it appropriately, and maybe when it mounts out here to the surface, it doesn't stick right or something. Right? So let's talk about the lysosomes. I've already covered this, but the lysosome is full of hydrolytic digestive enzymes. It's like the stomach of the cell. It eats bacteria, it eats debris you don't want. So like, for instance, a food product can come in here. This food product comes in, the lysosome comes up to it and makes that thing called the phagolysosome, or a phagosome. Right? Digests the food, eats what it wants, absorbs what it wants, and spits the rest out. Here's another one. So if you have an old worn-out organelle, like a mitochondria, you package the mitochondria, you bring it over to the lysosome digests the mitochondria, and you spit out the waste products. It's a really cool process. Right? So here you can see and in this example, it's a peroxisome fragment that's in here, and a mitochondria fragment, and then you're introducing these things together, and you're just destroying that mitochondria. So lysosomes, I already kind of covered the characteristics you need, but here are some major purposes. It fuses with other vessels, and it does digestion, so it can break down all the macromolecules, fats, lipids, which are the same thing, proteins and carbs, fats, proteins, and carbs. Autophagy, which means it can eat the cell. It can eat the own cell. If a cell is ready to die because it's getting old and worn out, what will happen is that it'll release the lysosomes. It'll open them up. That would be like dumping your stomach all over the surface of your skin. Your stomach's so full of enzymes and acids that when you eat a piece of meat, it digests those pieces of meat. It'll digest you. When you open the lysosome inside the cell, it will digest all the inside of the cell. It'll just eat the whole cell from the inside out. Okay, so it's self-eating. Autolysis means it destroys the cell. It'll break the membranes and pop the cell. And then it's also there for detoxification. By the way, I forgot to add colors to this. You definitely have to know a lysosome, so that should be in red, and then you should know these things in red. These are the main purposes. Next are the peroxisomes. So peroxisomes are just like a lysosome, except they start with things called free radicals. And these free radicals are destructive. They're dangerous. Like one free radical is called hydrogen peroxide. Where have you heard of that before? You buy it in a store. It comes in a brown bottle. If you put that on your hair, what's it do to your hair? It strips the pigments out of the hair and changes the shape of the hair. So it's very toxic. Well, your body makes these things. Your cells make these things as a protective mechanism. It's almost like a bee makes venom to protect itself, or a snake makes venom to protect itself. Well, your cells make this peroxisome to protect itself. So things like white blood cells will make peroxisomes that they use to destroy bacteria. Right? They can spit some of these free radicals out on the bacteria, and we'll get to the immune system. We'll talk about that a little bit more in detail. So instead of hydrolytic enzymes like a lysosome have, it has oxidase enzymes. So it oxidizes things, and those enzymes produce free radicals. And the free radicals can break down amino acids, they can break down mitochondria, they can break down toxins like alcohol, and then the final product, when you've made this peroxide, once you've neutralized it, it makes it into water and oxygen. Like I said again, it's kind of the green organelle. It's very safe to use. Right, so here's a direct comparison of lysosomes and peroxisomes. They're both vesicles, but the lysosomes, when they're made, they bleb off of the Golgi. Peroxisomes technically self-replicate. They make themselves, so they copy themselves to release. The lysosomes use water-loving components to break things down, where the peroxisomes use H2O2, which is hydrogen peroxide, to break down things. These have a tendency to break down all the macromolecules. These have a tendency to just break down fats and toxins. So here are some comparisons. And the next one's called a vault. And vaults are kind of, well, they're relatively new to being discovered. The vaults look like symbols to me, but they're special structures. They're like a vesicle, except they're more for isolation. So let's say you have a toxin that comes in. Instead of just putting it in a vesicle where it could potentially leak out, you put it in this vault, and the vault locks around it and turns into this barrel-shaped structure so that nothing can get out. Right? What they found about vaults is that it seems like the people that have higher resistance to chemotherapeutic agents actually have more vaults. So what's happening is that chemotherapeutic agent gets in the cell, and the cell isolates that dangerous poison. Right? So again, the endomembrane system involves all of these membrane-bound vesicles, or membrane-bound structures, organelles, from the rough endoplasmic reticulum all the way to the vesicle. And they help do things like produce, store, and export molecules, as well as break things down. The last organelle we're going to talk about is really important. In fact, I'll have another video that talks about what's going on inside this organelle, but I'm going to summarize it right now, and it's called the mitochondria. The first thing you want to notice about the mitochondria is it's not just a membrane-bound organelle, it's a double membrane-bound organelle. So you have an outer membrane, and you have an inner membrane. Right? The inner membrane, they call these cristae, going back and forth, these weaves. Usually when you see it drawn, you see the zigzag line going through. But these inner membranes have enzymes that break down food components. And when you probably in biology learn this, but there's special processes called the Krebs cycle and the electron transport chain. And what they do is they take electrons. So we talked about atoms briefly in physiology. But they take electrons and bounce them around to manipulate the energy of the electron. So here's the big overview. On this side, oxygen, ADP, not ATP, this is a precursor to ATP, and nutrients come into the mitochondria. It manipulates these things, processes them, moves them along the cristae, and on the other side pushes out ATP and carbon dioxide. It makes a lot of energy and then this waste gas. This is kind of the same product that your car goes through. Your car breathes oxygen, whether you want to believe it or not. It needs to breathe. So it brings in oxygen. It also brings in its nutrients, gasoline, sends it through combustion chambers, and then poof, cranks out energy and heat and carbon dioxide out of your tailpipe. Right? The big point with the mitochondria is it makes lots of ATP. ATP is like the money of the cell. It's the currency of the cell. If you want energy, you got to have ATP. Okay? Here again is another picture. Here you can see an electron microscope picture of the mitochondria. You can see all the cristae going back and forth. Right? We usually consider it the nuclear power plant of the cell. When you put one piece of energy substance like a sugar, when you take one piece of sugar and split it apart and give it to the mitochondria, this mitochondria, that one piece of sugar, can actually make about $36 worth of ATP, so 36 pieces of ATP. Other processes of making energy are not nearly as efficient. So we call this a nuclear power plant because it creates an abundance of energy. Right? It generates most of the cell's energy. It's the primary form that the cell makes energy in the human body. Right? And of course, again, anywhere that you find a cell that needs a lot of energy, like a sperm that has to swim for potentially 3 days to find an egg, it needs lots of mitochondria. Your brain that requires lots of energy, lots of mitochondria. Your muscle cells that require a lot of force and energy, mitochondria. It's a very complex organelle. In fact, they think this thing was alive outside of you. They believe that evolutionarily, it was a bacteria that was so good at making energy that when your cell ate it, it goes, hey, you know what? You're pretty handy. I'm going to keep you around instead of destroying you. And so we've actually been using this mitochondria that was at one time a bacteria to keep us alive. And it's kind of interesting because mitochondria are actually not made by your cell. Mitochondria make themselves. They replicate themselves. So we'll talk about this a little bit in reproduction, but your mitochondria inside of you is all from your mother. It's the same mitochondria that your mother had in her egg cell that she donated to make you. So that mitochondria just keeps replicating. When you get mitochondria, you always get it from your mother. Another cool thing about mitochondria is it actually has DNA in it. It has its own DNA that's separate from the DNA in the rest of the cell. So this DNA just makes products for the mitochondria. That DNA in here came from your mother. So we can actually track your maternal lineage, your mother's side of the family, way back looking at just your mitochondria, because all the DNA in the mitochondria is from mom. Okay, so how cells transform matter. We're going to talk about this more in digestion, but there are two terms you need to get familiar with now. Catabolism and anabolism. Catabolism means you take some kind of a nutrient and you break it apart. You destroy that nutrient and you release its energy. So when you eat a piece of sugar, that sugar has stored energy. You catabolize it and you release the energy. Right? So you're breaking it down. I always think catabolism is catastrophic. It's destroying things and all this energy is released. So when you store fat or you store sugar in your body, when you want that energy later, you just break it down and use the energy. Right? Another process is called anabolism. Anabolism, I always think of anabolic steroids, which build up muscle. Anabolism makes large molecules. It takes energy and stores it in the large molecules. Okay? So it requires a little bit of energy because you put the energy into that and that energy is stored. It's like putting money into the bank. You store the money in the bank, in a large bank, and then when you need it later, you can go and take it out and break it down. Right? So this is a process. Catabolism is accessing or releasing stored energy. Anabolism is storing that energy for later use. And here you can see this pathway. So you have these, well, we'll start with this complex molecule like a carbohydrate, a starch. You break it down during catabolism, you release some heat, and you release some energy. Well, whatever's left over, you can store it for later through anabolism, so that when you need it later, you just re-release it, break it down, and use it. All of these metabolic pathways require enzymes, and this I have a whole video on it that talks about enzymes and the metabolic products and things, but you need to remember enzymes are there to help speed up reactions. It helps things happen. So we have these enzymes. We'll take something like a substrate. We'll say this is a sugar, and this is an enzyme. That substrate goes into the enzyme. The enzyme takes that substrate and splits it apart into products. So let's say this is lactose. Here you have lactose coming into this enzyme called lactase. Usually enzymes are labeled ASE at the end. And it splits it apart into two smaller sugar particles and releases them. That's what enzymes do. Right? Food requires enzymes to be turned into ATP. And you actually have enzymes that process each of these. You eat the food, you have enzymes that break the food into small, tiny particles. Once you have the small, tiny particles, you bring them into the cell. Enzymes go through a process called glycolysis or oxidative phosphorylation. So we'll talk about these just briefly, but there's a whole video that talks about this process. So enzymes help speed up this reaction. That substance goes into the mitochondria, goes back and forth across the cisternae, or cristae, I'm sorry, and then pushes the final product out. It's a three-step process for you to actually take food and break it down to ATP that's usable. And here's an example. Aerobic pathways using the mitochondria means that you have to have these things, have to, and that's why they're in red. You have to have oxygen, and you have to have a food source. So you can have fat if you want. You can take the fat, break it down to something called glycerol, and into pyruvate. You can take a glycogen, like sugar, carbohydrate, like glucose, and break it down to pyruvate. You can take a protein, break it down to an amino acid, which can be turned into pyruvate, and also something called ammonia and urea. Waste products, kind of toxic. All the pathways come to this pyruvate, and then they go into the mitochondria in the presence of oxygen to make, altogether, roughly 36 ATP. You can use any of these food sources. The cleanest food source is glycogen. You take glycogen, turn it to glucose, pyruvate, and you burn it and make CO2. Fatty acids go into this product to make waste products. Proteins go into this and make other waste products. Fats are high in nutrients, but they're kind of nasty when you go to burn them because they're so full of waste products. And we'll talk about that later in a different video. Right? So when you're burning energy, or making energy, I should say, making energy, you have two major processes. You have oxidative phosphorylation, which happens in the mitochondria. One piece of food source will make 36 ATP, some water, and carbon dioxide. The alternative, if you don't have oxygen, remember you have to have oxygen here. If you don't have oxygen, you can use a process called glycolysis. It's pronounced glycolysis, but look at it. Glyco is referring to the sugar. Lysis means to break it apart. Right? So this glycolysis happens in the cytoplasm, but it only makes 2 ATP, 2 pyruvate molecules, and it also makes 2 lactic acids in the final product. So that's going to make that toxin, lactic acid, that you always associate with muscle soreness. And this is what glycolysis looks like. So glycolysis, you take the sugar, pyruvate, turn it into lactic acid. No mitochondria involved, but you only make $2 worth of ATP from that sugar. If we go back a step and we look, here's that one piece of sugar turned to 2-pyruvate and fed to the mitochondria. The mitochondria takes oxygen, turns that glucose into about 36 ATP altogether. Huge difference. The mitochondria is more efficient. It's the nuclear powerhouse, where glycolysis is very wasteful. When you get into physiology, we start talking in physiology about metabolism, like in the muscle. We'll start talking more about this glycolysis and how mitochondria work too. But right now, this is just the big picture. Compare glycolysis to oxidative phosphorylation and mitochondria and what they need to be able to make energy. Right? So big picture, you can take any of these food substances, right? You can do anaerobic without oxygen, and you can make a little tiny bit of ATP and then that bad lactic acid. If you have oxygen, just a little bit of oxygen, you go through an aerobic pathway that makes lots and lots of ATP, and the byproduct is CO2, which you just blow out. Right? So take a little time, go through this and review. Just kind of look at all these structures that are inside the cell, and can you name each of them? Go back and look at your notes. See if you can name these little crisscross microtubules. See if you can name this thing with the cristae going back and forth. See if you can name these different structures in here. Where do you usually find them? Like when we talked about this rough structure, it was directly attached to something else. When we talked about this, well, this thing around the outside of the cell, it's always around the outside of the cell, but what's its name? What's its function? So see if you can go through that, and then I also put a link to something I thought was really kind of cool. It's called The Owner's Guide to the Cell. Right? And again, if you get a chance, Google the inner life of the cell and watch the video.